All right, you're in the right place, and if you're interested in following along at home, there's a link to download our entire validation report. I'm Ramji Srinivasan, co-founder and CEO of TACO, and our mission is to make high-parameter or multi-parameter cytometry for clinical trials routine and simple. And today we're going to talk about our CLIA validation for our 25-marker spectral flow test on fixed whole blood. So, why are we here? Because high-stakes measurements in immunotherapy trials need high reproducibility. Now, assuming you're spending $5 million on a 12-month drug program with 8 enrolled patients. 5 of them get cured, 3 of them didn't. Now the question is, did the cure happen by magic or did it happen by science? So say you use tools like cytometry to analyze the patients and compare responders and non-responders. If your cytometry tool has a very wide coefficient of variation, also known as CV, you might get a chart that looks like this. Here you have responders and non-responders by response group and the percent of a top-level immune population. And if you're looking at this graph, you probably wouldn't bet another $20 million on another phase 2 just on this kind of separation. But if you had tighter CVs, you might be able to reveal the underlying biology and can see clear separation between responders and non-responders. So now the question becomes, how do you get those low CVs? There are 5 big sources of variability: instruments, reagents, specimens, operators, and controls. And today we're going to talk about how blood specimens can help dramatically reduce the variability in your trial. So there's a big dilemma for immunotherapy developers, which is they have 2 bad options. One is to use what's called PBMCs, or peripheral blood mononuclear cells, on trials. And these are very expensive, they miss granulocytes, and they have high variability in viability. The other option is to try to draw everything live and race to a lab within 72 hours. And if you've got Europe or Asia, or even within the US trial sites, there's still a 10 to 20% failure rate. And when you drill into live samples or fresh samples, we can see that there is actually really big degradation. Not every fresh sample is created equal. This is a sample at 0 hours compared to sample at 24, 48, and 72 hours. This is TACO original data. On the x-axis, you can see time from 0 to 72. On the y-axis, you can see the cell count, percent normalized to t equals 0. And across these 3 donors, you can see 50% of cells are dead by 72 hours. Yet live, no matter if it's in 0 to 72, is considered equivalent. Now, fixation is a tool that could potentially solve this issue, but it hasn't yet been optimized for multi-parameter cytometry. And that's what we set out to do with our TOCO kit. This is a collection process. You can collect blood in a vacutainer at the clinical site, invert vacutainer 10 times. You transfer 2 ml of blood to buffer 1 and incubate for 15 minutes. Transfer that entire mixture to buffer 2, and then store at -80 or ship on dry ice. So this is a great way to fix samples, and we're going to talk through how we've paired this with spectral flow to create a great reliable assay for trials. So more details on what we tested. We tested a large 25 marker spectral flow panel. So we started all the way at CD45, going down to CD127, CD28, CD66B, covering all the major cell lineages: T, B, NK, monocyte, dendritic cell, and so on. Here's a quick snapshot of that entire cell lineage and functional markers. You can see the leukocytes over here, the T cells here, CD4 T cells and all the different lineages, CD8 T cells, the natural killer cells, dendritic cells, monocytes, and B cells. And the gating charts are all online if you're interested in drilling down in more detail. Within each cell type, we also measure functional markers. So on the left side, you can see all these different functional markers, and you can see where we measure those particular functional markers in. So for example, LOX1 we measure only in monocytes. CD15 we measure in granulocytes and monocytes, for example. What we did in our test was we used whole blood fixed with the TOCO kit from 3 healthy donors. So we took 3 healthy donors, fixed them with our TOCO kit collection, and then analyzed on our spectral cytometer. We have an Aurora spectral machine made by Cytec. And now the drug developer's question is this test valid? And so we had set out a validation plan which had 4 measures: limit of blank, intra-run precision, inter-run precision, and stability. And for each of these, we have a specific meaning. So, for example, limit of blank, you take that marker out and make sure that it doesn't show up when you run the actual test. Inter-run precision, same sample, different runs. Stability, same sample across time. And there are a variety of different acceptance criteria depending on it. And we're going to talk about the phenotypic subsets, analyze the phenotypic readouts and the phenotypic population below criteria. We do have the functional data. We will show that as well, but in terms of the criteria, we really focus on the phenotypic ones because these have a little bit less variability. So let's take limited blank. As a reminder, this is based on fluorescence-minus-multiple FMM controls design. We have a video on that if you're interested as well. We start with a normal blood sample with 25 marker panel. Then we take out 5 markers, call it FMM1, fluorescence-minus-multiple 1. We take out another 6 markers, not the same 5 markers, FMM2. Then we go to FMM3, which is another 5 markers, not covered in 5 or 6, and FMM4, which is another 5 markers, not covered in these 3. To see that laid out, you can see the actual panel over here, all 25 markers, FMM1 with those markers subtracted out, similarly for FMM2, for FMM3, and FMM4. Basically what's going on underneath the hood is, for example, in FMM1, CD141 doesn't appear on this particular panel, so if you run this sample, you want to make sure that this doesn't show up in FMM1. Similarly for CD16, it ought not to show up in FMM2. So lower numbers are going to be better. Here are the results. So across all these markers, you have the percent marker positive in the full panel. These numbers ought to be high. And then the percent marker positive in the corresponding FMM. Remember, lower numbers are good here, and the threshold is 1 or 1%. So 0.041 all the way down to 0.16 compared to the 1 threshold. If you see this plotted out, you can see CD1C all the way to PD and the percent marker positive, the acceptance limit of 1% over here. So again, great results. So this is the first notch on our belt, which is limit of blank. Instead of a 1% event criteria, we were all the way down at 0.081%, and 100 populations were below criteria. And we have these other measures as well, intra-run and inter-run precision and stability, so we're going to cover those next. So let's go to intra-run. This is the same sample, same run. So each donor sample is split into 3 replicates, and then we run that on the spectral cytometer, and then the goal is to ensure the results within a run are consistent under the same conditions. So here we have by cell population size. So the median percent CV across all 3 donors is 3.38%, and this is for phenotypic markers, and for the functional markers, the median percent CV across all 3 donors is 5.50%. So as we look across the intra-run precision compared to the CV of less than or equal to 20%, we notch 3.38%. So great performance there. Next, we're going to cover inter-run precision and stability. This is the same sample split into 3 replicates. So you take the blood sample, split it into day 1, day 2, day 3, and the goal is to ensure results are reproducible across multiple trials. So here's the precision by cell population size. These are the phenotypic side, this is the functional marker side. You can see the median percent CV across all 3 donors is 4.56. You can see over here, this is, for example, the 3rd donor, the 2nd donor, the 1st donor. For the 1st donor, you can see over here all the different population lineages, and this red line over here is acceptance criteria. Similarly here for donor 1, 2, and 3, you can see all the individual markers here, functional markers, and here's the acceptance criteria. So for inter-run precision, the CV of less than or equal to 25%, we found a 4.56% average criteria readout. So that again passed. And last is stability. So here we compared fixed blood at T equals 0, 1, 12, and 24 months. And you notice 12 and 24 months are accelerated, and we'll talk a little bit about that. Each donor we split into 4 replicates and compared at each time point. So here's a percent change from baseline at 1, 12, and 24 months. So 1 month we saw 6.51%, 12 months is 5.2, 24 months via accelerated 7.74. And again, over here you can see all the individual populations colored by lineage. Here are the functional markers, CD28 to PD1, and then for 1 month, 12 month, and 24 month. So 1 month 8.36, 12 months 8%, 24 months 12.94%. So we talked about accelerated stability, and the American Society for Testing Materials has defined a protocol for accelerating the aging of samples. This allows us to forecast the stability at 12 to 24 months while we wait to test our naturally aging samples. This is an underlying formula. We're not going to go through that in great detail. Suffice it to say that 12 months at -80 is comparable to 12 days at -30, and 24 months at -80 is comparable to 23 days at -30. So this is our validation plan. Stability, same sample across time, change plus or minus less than 25%, and we found it was 6.51, so well below the 25% threshold at the 1 month mark, and we'll continue to read out the naturally aging results. You saw the accelerated stability results as well. So the question from the drug developer is this test valid? And the answer is yes. In terms of the validation criteria, we found our test to meet or exceed. The one asterisk here is on stability. We saw plasmablasts and transitional dendritic cells Those showed high variability in the stability tests, and excluding those, we would pass this plus or minus 25% mark across more than 95% of populations. So the answer is yes. For our validation plan, we have these 4 measures, and you can see that we have set out these criteria and handedly exceeded them from limit of blank to intra-run precision to inter-run precision and stability. So if you're interested in reading further, check us out at taco.bio/whole-blood-spectral-flow. Thanks for signing on.